on how strong the particular elicit an what do you call an impulse in the postsynaptic junction, and all these neurotransmitters and neuromodulators are actually they actually modulate the transmission of the symbol. It may actually start getting depolarized until the action potential is generated. Okay, but in that process, if a neuromodulator arrives from somewhere else, and that may actually increase the depolarization, or it may decrease the depolarization. That is, a neuromodulator can be, what you call excitatory or inhibitory or something like that. There are so many neuromodulators identified. Some of them may be amino acids and other types of chemicals, and sometimes gases, etc. Okay, those are modulators. Okay, they may increase or decrease the excitability of the neurons, and so they are called neuromodulators, and there are so many of them. Okay, so now coming back to that, see the importance of the interneurons, and this can happen. There are signs. Plastic means they can keep on changing. Chemicals and new synapses may form, and existing synapses may get removed, or the synapses may keep getting strengthened. That is, the area of the synaptic junction can become broader and broader, whereby a large quantity of neurotransmitters may be transmitted from the presynaptic to the postsynaptic, that if there is a strong synapse. So now these are all So this is what keeps on happening, and this is how we actually, strengthen our memory. The network, and this network is not static, and it is dynamic. It keeps on forming and changing itself. I always keep giving an example of a newly formed large colony with a lot of roads. Width and equally well built and all those things, but over a course of years there will be other roads in the colony which are not frequently used. So gradually the well better maintained, and they may become wider and all those things. This is what happens even in neural networks. If there are preferential channels, particular parts of the network may keep getting stimulated more frequently, and that those synapses in that particular route will become more stronger and stronger. What happens if the synapses between the rest of the network may get withdrawn, and they may get removed. So this is the kind of plasticity we have in the synapses. Now, and you get certain kind of, if a subpopulation of neurons in the network gets stimulated, then you will get a certain kind of a memory. And if that particular subset, you keep losing the details of that memory. Okay, so now this so-called memory to remember the outcomes which has given advantage to us, and then if you come across a similar situation, right? So now all this resides in the synapses. Okay, so now is it okay? Now more number of interneurons will lead to more complex behavior, whereas step on a thorn, you just withdraw. There is no other kind of behavior, or you need to press it harder against that particular thorn. You don't do that, right? So there is no need for a complex behavior for that kind of. So these are all reflex between the dorsal horn and the receives the sensory inputs, and the ventral horn gives out the different output to the muscles or glands or whatever it is. Now, there are various types of behavior. No, the behavior may involve muscle contraction. It may involve the production of sound through the respiratory and the laryngeal muscles, or it may be secretion from some glands or whatever kinds of things. Okay, or it may be kind of through the efferents, ultimately executed through the efferents. Okay, so now these interneurons will keep on get accumulated in groups of cells, which can actually get organized in what are known as the basal ganglia of neurons, which appear in the evolution in the so-called whatever is appropriate behavior is actually output from this cortex, and then the lower centers will actually transmit those resultant in system, which may be the motor centers, or which may be secretory or whatever kind organized. Now, have you got some idea of what I was trying to convey? So now, if there are more and more, then the more and more complex patterns of behavior can be planned and executed in the interest of the preservation of the individual. Okay, now is that clear to all of you? And try to see the various parts of the human nervous system. So, like all the other systems, the nervous system is also made up of cells. Okay, the primary cells of the nervous system are called neurons. And then it has processes which are known as the neurites. Neurites can be axons or dendrites. Generally, the axons carry the impulse from the peripheral part towards the body of the neuron, and the dendrites, sorry, the other way around. The axon carries the neural impulse from the body to the from the cells, from other cells surrounding them through the axons, and the dendrites will convey the impulse towards the cell body. Okay, and now where these axons and dendrites of a particular neuron may end up on the body of another neuron, and then ends up on the dendrites of the other neurons. Okay, and they are called the axodendritic nervous system are called the neuroglia. Glia actually means glue, or they are named as neuroglia, and neuroglia are the supporting cells, and there are various types of neuroglia, and we will be just doing the details. You will go into details of these neurons and glia in subsequent. Actually carry the impulses towards the muscles, which actually stimulate the muscles, and those are a kind of synapse between the neuron, the axon, and the muscles, and those are called the neuromuscular junctions, and you will also see learn about the details of this. And here you can see the neuronal cell body, and these are all various organelles nucleus of the neuron, and this long process is the axon, which goes and synapses with the target organs like a muscle or that is the axon. And axon in many axons which require a very fast around the axons, and then there are some gaps between the myelin sheath is segmented because each segment is actually provided by one particular Schwann cell, and then there are some the so-called nodes between the segments of the myelin sheath, and those are called the nodes of Ranvier, and the synapses generally branch out before ending on the target organs. Okay, so this is an axon, and these are the dendrites, and the dendrites have a large number of projections known as synapse with them. So these the dendritic spines, the dendrites are highly branched, unlike the axons. Axons are terminally branched into few branches here, whereas dendrites have a large number of branches, and then there are dendritic spinous processes on that. So this is a schematic representation of a neuron, and each one is a dendrite here. This is a dendrite. Okay. So the real neurons, which are Golgi stained, this is a particular type of staining technique which actually deposits opaque salts or processes of neurons. Okay, and this staining is called Golgi staining, and these are some of the neurons known as the pyramidal cells in the cerebellum most probably, and these are all the dendrites of these particular tree. Okay, the dendritic tree. So this is how the neurons look in Golgi stained preparations. And these are all neural cells, and you can see that these are all multipolar neurons, and there are neurons where a single process comes out of the cell, and then it divides into a peripheral process and a central process. The peripheral process brings the impulse, takes the impulse away from the cell body, and those are the pseudounipolar, and these are found in the sensory ganglia attached to the dorsal roots of the spinal cord. Okay, so these are the neural cells. Again, these are the various types of neurons, bipolar. So now I have all part of the nervous system which gathers information from the world, which may be outside world as well as the environment inside the human body. Also, information has to be gathered about, for example, the fullness of the urinary bladder or emptiness or whatever kind of things. So the motor, or they can even be secretomotor, secreto if they are going to the glands. The network which is actually directly not connected with the outside world or not directly giving out, but it is actually processing the information and then handing over to the efferent limb. By the genetic organization, the nervous system, the patterns of the networking in the nervous system, and then they may also be environmentally influenced. response to new experiences or whatever kind of things. And then if we don't come across such an experience again and again, we tend to forget it. So now, that is called the plasticity, and it is influenced by the environment. So genetics are determining the basic patterns of the networks in the nervous system. Patterns of the networks of the neurons. So if you see the development, you know that a zygote which becomes trilaminar and all those things. I hope you are aware of that. Okay, and when it is trilaminar, the names given to the layers are the ectoderm, endoderm. The endoderm gives rise to the gastrointestinal tract and all the derivatives of that, and mesoderm gives rise to most of the connective tissues of the whole of the body. Okay. The germ disc is somewhat like a racket shaped, or tube is the midline part of the ectoderm, which actually transforms into the so-called neural tube, and that tube gives rise to the brain vesicles of the brain. End of the neural tube, and those are called the brain vesicles. And then where the neural towards the edge of which does not get incorporated into the neural tube or into the ectoderm is called the neural crest. In the embryology of the nervous system. Tube. Okay, this side is the ventral side of the neural tube, and this side is the dorsal side of the neural tube. So in the region of the fourth ventricle, that is called the rhombencephalon. The prosencephalon, that is the forebrain, and then the mesencephalon or the midbrain, and the rhombencephalon or the hindbrain. Okay, and this is a cross section of the rhombencephalon. And in the wall of the neural, and these are all different columns, and these columns end up innervating different parts of functionalities. That is why they are called functional columns, because each column subserves a particular function. Sensation, somatic sensations, and that is this inner part where there is not much of cellularity is called. Okay, and then there are all these somatic and visceral. This part of the neural tube, that is the dorsal half of the neural tube, innervates the body wall and the related. Is the ventral half of the neural tube, innervates the viscera, and particularly those afferents, and this is efferents, and the ones which are near to the middle part of this thing, they innervate the viscera, whereas the parts of the neural tube which are away from this particular groove along they innervate the body wall or the somatic columns, and these ones closer to this are the they innervate the viscera. So the dorsal half is sensory, this whole thing, and this is are concerned with the viscera. So, and here we have somatic particular sulcus limitans. Can you see this groove here? That is called the sulcus limitans. Closest to the sulcus limitans, we have the columns which are concerned with innervation, afferent or efferent of the viscera. So here on this side we have the afferents to the viscera, whereas here we have efferents to the viscera. Okay, so the human nervous system is divided into central nervous system and peripheral nervous system. So the brain and the spinal cord are considered to be the central nervous system, whereas all the other either connected to the cranial nerves or the spinal nerves, that is, the whole thing is called the peripheral. Sympathetic and parasympathetic, and then in the wall of the gut there is a large collection of neurons forming very extensive network which extends throughout the length of the gastrointestinal system, and that is called the enteric nervous system. So which are connected to the cranial nerves, that is connected to the brain. The third one, which is in the walls of the gastrointestinal system and the related glands, etc. of the GI tract or the enteric nervous system. Cover the nervous system are called the meninges, and these meninges, there are three layers of these meninges. The matter, because it resembles a cobweb. You know, arachnida is this thing class to which the spiders belong, right? So this is called arachnoid matter, and pia matter is the thinnest of the layers, and it is intimately adherent to the surface of the brain and the spinal cord. So that is the pia matter, and these are some of the pictures that I'll show you. And this is the cranial wall, and on the surface of the brain, closely adherent to the surface, and there is a capillary space between the dura mater and the arachnoid matter. Between the pia mater and the arachnoid matter, which has got lot of septa and subarachnoid space, and cerebrospinal fluid fills this subarachnoid space. Okay, and this whole thing is subarachnoid space, and the blood vessels on the surface of the brain are in the subarachnoid space. This is the falx cerebri hanging here, and these are the venous sinuses, dural venous sinuses here, and this is actually here, and this is the grey matter deep inside the cerebral hemisphere, and this is the grey the neurites, that is the axons and the dendrites of all these cells. These are the dural venous sinuses here, and this is called superior sagittal sinus, and this is like sickle-shaped projection of the dura mater between the two cerebral hemispheres. And these are the venous sinuses which are there in the folds of the dura mater. They are known as the cavernous sinuses here, and then you will see the details of this. You know, this is the transverse sinus. So of this, and these are the cranial nerves which are exiting through the foraminae inside the cranial nerves. The fifth cranial nerve, that is the trigeminal nerve, it gives rise to three branches. That is why it is called trigeminal, and this is the optic nerve getting attached to the eyeball here, okay, which innervates the retina. this tent is the cerebellum, and above that is the cerebral hemisphere, the occipital lobes of the cerebral hemisphere. So I already mentioned that the neural tube gives rise to three vesicles or the swellings, and those are called the prosencephalon, mesencephalon, and rhombencephalon. Hindbrain. The prosencephalon gives rise to the cerebral hemispheres and some of the parts of the brain which are between the two cerebral hemispheres. Interbrain or the diencephalon. And the cerebral hemispheres themselves are derivatives of the telencephalic vesicles, which come out from the prosencephalon on either. The interbrain, that is the thalamus, hypothalamus, and some other smaller structures. Mesencephalon gives rise to the midbrain. Rhombencephalon gives rise to the cerebellum and the pons. That these two together are called the metencephalon, and the medulla oblongata is called the myelencephalon. And below the medulla oblongata we have the spinal cord. That is, it is a derivative of the rest of the. Okay. So here you can see that up to this part the neural tube is divided into three. Sorry, this is the mesencephalon, and this whole thing is the prosencephalon, and the red part is the diencephalon. So this is the mesencephalon, it gives rise to midbrain, and this is the rhombencephalon. Rhombencephalon is void. It is diamond shaped, which ultimately gives rise to the fourth ventricle of the brain. So that is why it is called rhombencephalon. And this rhombencephalon gives rise to cerebellum and the medulla oblongata. Okay. Telencephalic vesicles give rise to cerebral hemispheres, and this red part gives rise to this. And this purple part gives rise to the midbrain here. Now midbrain. Then the cavities, this cavity in the telencephalic vesicles gives rise to of this diencephalon becomes the third ventricle. And this aqueduct is actually connecting the third ventricle, that is this part, with the fourth ventricle. This is the fourth ventricle here, and this is that floor of the fourth ventricle, which ultimately becomes the cavity of the midbrain, which connects the third ventricle with the fourth ventricle. Here you can see this is the fourth. The center of the spinal cord also there is a canal, and that is called the central canal of the spinal cord. And caudally, the fourth ventricle is continuous with the central canal of the spinal cord. Okay, these are the cavities, and then the shape of the fourth ventricle is like this. It has got horns are in the occipital lobe, and inferior horns are in the temporal lobe of the lateral ventricle. And the lateral ventricles are connected with the third ventricle through the so-called interventricular foramen. This is the roof of the fourth ventricle. Here. That is the frontal lobe. And this part is the occipital lobe, this part is the temporal lobe, and this part lies over the tentorium, and this part lies over the orbit, over the roof of the orbit. And this is the superolateral surface and the project sulcus sulci, and this is called the lateral sulcus. And parietal above and the temporal lobe below. This is the lateral sulcus. You see the detail. So this blue part is the parietal lobe, and this is the occipital lobe, and this is the temporal lobe, and this is the frontal. If you open the insula actually refers to an island. Okay, this is deep inside the lateral sulcus, and you can see that the blood vessels are there on the surface of this cerebral hemisphere, and their branches pierce the cortex and supply the cortex. This is actually the middle cerebral artery, which is in the lateral. This is the cut part of the midbrain, and this is the aqueduct of the midbrain, and these are the this is the under surface of the frontal. This is the under surface. The two cerebral hemispheres are connected by what is known as the corpus callosum. And this olfactory bulb lies over the roof of the nasal cavity, and there is a sieve-like bone in the roof of the nasal cavity. The other parts we will just study later. Cerebral hemispheres. Okay, similar areas of both the cerebral hemispheres are connected through the corpus callosum. Okay, and this region is the thalamus, and this is the cerebellum here, and this is the roof. Is the midbrain. Okay, so this is pons, and this is the medulla oblongata. These two together are called the metencephalon, and this is called the myelencephalon. Okay. Thalamus and epithalamus and all these kinds of pineal gland is here. The third ventricle, actually this foramen through which the lateral ventricle, which is deep inside the cerebral hemisphere, is actually opening into the third ventricle. Of Sylvius. Okay, so are the parts of the brain clear now to everyone? This is the medial surface of the cerebrum, and this is actually the anterior cerebral artery. Just some time back I showed you the branches of the middle cerebral artery in the lateral sulcus, and this is the anterior cerebral artery, which is actually traveling along, which is bending around the anterior end of the corpus. Large number of branches which supply most of the medial surface of the cerebral hemispheres. They are all supplied by middle anterior cerebral, and whereas the posterior parts, they are supplied by here. You see, this is the posterior cerebral artery, which is supplying the medial surface of the occipital lobe, and this is the area where the visual lobe, which goes up to the pole, occipital pole. Okay, so this is the pituitary hanging from the floor of the third ventricle. It is connected to the floor of the third ventricle. That is, this region is called the hypothalamus. So you know that the pituitary is connected to the hypothalamus, and this is the thalamus. I think this may look a bit complicated. Let us just postpone this until when you see the actual brain, the dissected parts becomes easy. This is actually in the rim surrounding the communicable which develop there, and that is called the hippocampus, because it looks the hippocampus, and these are all the bundles of fibers which are connecting. These are called the mammillary bodies, and this is called the hippocampus here. And in the previous slide you might have seen, this is a bundle, right? This bundle is coming down to the mammillary bodies here, and actually it is going like that, and then it is going towards the lateral side, and the hippocampus is on the medial surface of the cerebrum, which is lateral. Okay. This part, this arrow, if you pay attention, it is this. And see, it is going lateral face is called the hippocampus. The fibers, okay. So white matter is fibers, the gray matter is the cell bodies of the neurons, and this is corpus callosum. You can see that these are all parts of the cerebral cortex, right? These elevated parts are actually folds will increase the surface area of the cerebral cortex. So our brains, human compared to the other lower animals. If you have seen the brains of other lower animals, dogs and like the humans, so that increases the cerebral cortex, and that is where we have a large this. Now suppose this area on the left side of the cerebral cortex is connected to similar area on the right side of the cortex, and these are collections of neurons deep in the substance of the cerebral hemispheres. So now this is the white matter, that is the fiber. This is the deeper gray matter, and this is the surface gray matter. Surface gray matter is called the cortex, and these are all called the basal ganglia, and then there are names for that, you know. Different is the shape, the lens. So these are all further divided, and you will have fancy names. You will become familiar with these as you keep on using them. Okay, the globus, pallidus, vitamin, and all. And this is the this thing, thalamus, and this is called the caudate nucleus. Caudate because it has got a tail. You'll see in multiple sections, and that is a caudate nucleus, and this cavity. Now there are fibers which are connecting adjacent gyri. For example, this is one gyrus and this is another gyrus, and this is the sulcus between the two gyri. The neurons of this gyrus are connected to the neurons of the adjacent gyrus, and those are called the short association fibers. Now association fibers are the fibers which connect the centers on the same side of the cerebral end. So they and this is the occipital lobe. Okay, if there are fibers connecting the neurons in the frontal lobe with those in the occipital, are given to them. If they are looking like some sickle or something, they call it cingulum. So this is superior longitudinal fasciculus, inferior longitudinal. Inferior longitudinal fasciculus is connecting the temporal lobe here with the occipital lobe, and that is the inferior longitudinal fasciculus. I hope you are called longitudinal fasciculi, and fasciculus is singular and fasciculi is plural. So of fibers, uncinate fasciculus. So now these are all association fibers. Now do you understand my association fiber? Same cerebral hemisphere on the same side. So largest commissural commissure in the human body is the corpus callosum. Areas of one side cerebral hemisphere to the other side. So if the centers in the upper part of the nervous system are connecting to the lower or the midbrain to any lower parts, such fibers are called projection fibers. So, association fibers which connect in the same cerebral hemisphere, different areas are projection fiber. That is the centers in the higher upper parts of the central nervous system, starting from okay, these are all yes. Now see commissural. Projection fibers are connecting from higher centers to the lower centers, and these projection fibers may end up on the same side or they may also cross to the other side. Okay, so for example, root in the brainstem. Okay, so those are all called the projection fibers. Cerebral hemisphere, and those are called the basal ganglia. Get used to those names. So if we these are the basal ganglia. Can you see these collections? And this is the frontal pole, and this is the occipital pole, and this is the horizontal section. Okay, that means these structures, 3D reconstruction looks like this. We are looking at the looking from this side, from the left side. Okay, so this nucleus, the green part is the caudate nucleus, and this red part is the lentiform nucleus. Okay, so caudate nucleus, this is part of the caudate nucleus, and this is part of the caudate nucleus. Okay, so that means this section has gone like this. Have you got the orientation? This section has gone like to the lateral side. Okay, so here this side is you are looking from medial to lateral, and this part here is this part, and this part is the blue part. So okay, in sections, then it will become easy for you. Diencephalon has got various parts: epithalamus, thalamus, hypothalamus, and subthalamus. Epi is above the thalamus, and hypo is below the thalamus, and below the hypothalamus is the subthalamus. Is it the previous section, previous slide? Yes, somebody is saying ganglia, nuclear being used interchangeably. Yes, the definition is same. Now collections of outside the central nervous system also. Collections of neurons are called ganglia, nuclei. Okay, for example, the cranial nerves arise from neurons which are brainstem. Okay. Right. Thank you. Okay, now this is, maybe we can go to the previous slide and let us say, just to get the orientation, I'll just show you the cerebral hemisphere. Okay, now let's say, okay, and this is the medial surface that we are looking at. This is the anterior pole, this is the posterior pole. Okay, and this is the corpus callosum. And above the thalamus are called the epithalamus, pineal gland. Okay, now I will just try to draw a line across this, and this is the septum pellucidum, which is the line which I am drawing here. Okay, so this is the line along which the horizontal section is cut, which is shown in this picture. Okay, now let us go back to that slide and show you the fornix again. This is the fornix, and this is which are the cavities of the cerebral hemispheres. Okay, and this is the thalamus. Now if a section has gone